This is called Net Vitals, and some of you have seen this. Some of you have helped us develop it. This was something developed by Lisa Yen and myself, along with Dr. Dan Lee, as a way to help patients communicate with their physicians. And it's basically, we got this down into the fewest questions possible. If you take a look at the form, which is also available online, it's really the core questions for what define your disease state at this moment. So they're medical questions, and a lot of times we find that patients don't know the answers to all of these, and that's okay. That's actually why we did this, so that it's part of learning the same vocabulary, so when you meet with your physician, you actually are speaking the same language. And we did a webinar, which is available online. It's an hour long to take you through question by question. So if you have any questions about this, come speak with me or Lisa. Lisa, can you raise your hand? And we'd be happy to help you out with learning more about this. And the idea was that you bring this to your physician, and then your physician very easily can take a look at this and get a good sense initially of your disease state, rather than maybe bringing in a bank box full of your last few years of records. We also have NetConnect, which helps a program to help connect patients, and we have a new internal private email. And so if you want to ask someone, well, which do you like better, Sandostatin or Lanreotide? That's a great place to do that. What's your experience been with that? I'm having this procedure, and I'd like to talk to somebody else who had this procedure. The private email is a great place that's just with us. It's not open like our Facebook page. So that's a great way to network with other patients. So I'd like to introduce our speaker today. We actually have 3 speakers today. And Dr. Andrew Hendifar is the medical oncologist here at Cedars-Sinai. And he is here to speak along with Dr. Gong and Dr. Louise Thomson, who will be here shortly. So please welcome Dr. Hendifar.
Good afternoon, everyone. Thank you, LACNETS. Thank you, everyone, for coming today. I try not to bore you, so I invited 2 other speakers to kind of speak a little bit about some topics of interest. And everything we're going to talk about today are very, very current, very topical issues. I'll be talking about what's happening with immune therapy and neuroendocrine tumors. I think it's a topic that's very important. Dr. Gong was talking about precision medicine in neuroendocrine tumors. So there's different types of approaches to cancer therapy. One is immune therapy, one's precision therapy, and Dr. Gong will talk about that. And finally, Dr. Thomson will talk about PRRT, and she'll focus in on what are some of the newer ideas, newer things happening, not just for mid-gut tumors, but other types of tumors. She'll talk about how PRRT works for other types of patients. So let's talk about developing immune therapy for neuroendocrine tumors. So does anyone know who this is a picture of? So these are Nobel Prize winners for medicine, and actually 2 scientists who developed immune therapy for cancer. So we're very proud of both of them. I had the pleasure to meet Dr. Allison not too long ago. But they've done remarkable work, and that just shows you how important immune therapy is, that they already won the Nobel Prize for it. So it first started off, the story of immune therapy and cancer therapy started off for melanoma. And when I was in training, I was in the clinics actually when they were first giving IPI to patients with melanoma. And the most interesting thing about this story of immune therapy is that melanoma was a tough cancer. Really, no treatments really worked. And they were giving patients IPI, and one thing that happened was the side effects were different than any chemotherapy you've ever done before. I mean, completely different side effects. We didn't even know what we were seeing. Patients would have diarrhea, and we thought it's chemodiarrhea, but it wasn't. It was autoimmune diarrhea. And then they would get coughing, and we would think, oh, what's going on? You know, they have an infection. No, it's autoimmune inflammation, skin rashes, all kinds of new side effects that we're just all learning about. And the first few patients, they would tell us, we feel better. And we looked at the scan, and the scan wasn't that much better. And so we were very confused. And what ended up happening is we realized that when you give immune therapy, there's an immune flare. So at first, the immune system fights the tumor swells a little bit. Then later on, there's the shrinkage and the benefit. And what also different is that on immune therapy is one of the first cancer therapies that for some patients, not everybody, but for the patients who it works really well in, it can work for incredibly long periods of time. And there are still people on this study that are still, that are still kicking, still doing immune therapies. So melanomas became the prime treatment for immune therapy. Other therapies have been developed. So there's two basic classes. Both of them are what's called checkpoint inhibitors. I don't know if you've heard that word before. But the way checkpoint inhibitors work is basically by stimulating the T cells. Another way to give stimulated T cells is to actually develop them in the lab, and that's CAR T cell therapy. So T cells are collected from the patient's blood, they're reprogrammed to fight cancer and reintroduced. So the cool thing about immune therapy, again, is when it works, we want to see this. Some people, it works for a very, very long period of time, and you see separation of curves between standard treatment for how long a therapy works. Why does immune therapy not work for every cancer? So the issue is that for the immune system to identify a cancer cell, it has to look different than a normal cell, which is a problem for cancer, right? Because what is cancer? Cancer is just a messed up normal cell, right? It's a cancer, it's a mistaken cell, or I like to call them dumb. They're just kind of dumb cells. People say, give cancer nefarious characteristics. They're like maybe a bad child. Dumb. They're just kind of dumb, they make mistakes, they bumble around. And so if you don't have a lot of mistakes in the tumor, so if the tumor doesn't have a lot of mutations, it doesn't look disfigured enough, your immune system just can't see it. Now, does anyone want to get extra points and be really smart? Why is melanoma for the immune system to see? So two things. One is that melanin is something that the immune system can sometimes, you ever seen people who have patches of vitiligo? Have you heard of that? They have patches. So melanin is something that your immune system sometimes doesn't like. Not only that, sun damage. So sun damage causes a lot of inflammation, a lot of genetic changes. So the melanoma cells, when they're developed, the cancers, are quite disfigured. So the immune system already is able to kind of find them. So you're giving them a boost. So when you look across different types of cancers, immune therapies tend to work better when you have these disfigured cells. How does that work for neuroendocrine tumors? So based on what I told you about the immune system, can anyone guess, would it work better for high-grade tumors or low-grade tumors? The immune system works better for high-grade tumors? Yeah, you meant high-grade, right. Yeah, you did. Yes, right, because low-grade tumors are bland genetically, right? They don't have a lot of mutations, they're not disfigured, they're well differentiated. That's what that means. That means that they look like our own cells. They don't look so different. Versus the high-grade tumors, they're disfigured, they're high-grade, they have lots of mutations. And so that's the issue at hand. So this is looking at well-differentiated neuroendocrine tumors, PNETs, and carcinoids. This is one of the checkmate. It's a checkmate study. This is the first published data on neuroendocrine tumors. And in well-differentiated tumors, now these are investigator-reported, so investigators, we tend to be very optimistic and like, yeah, you're doing great, it's working, everything is, So it showed some promise in the beginning. Okay, so a carcinoid is an old term to mean a well-differentiated midgut tumor. A PNET is a pancreatic neuroendocrine tumor. So these are both well-differentiated tumors. One is from the pancreas, and one is from the midgut. Okay. Does that make sense? Sorry about that slide. Carcinoid is midgut? Yeah. Technically. But it could be, we use carcinoid freely to call anything. We can call anything carcinoid too. You can call a lung carcinoid, a pancreas carcinoid, a neuroendocrine. So some people use it interchangeably for neuroendocrine tumors. Yeah. Okay. So because of that early interesting data for well-differentiated tumors, what's another treatment that we have for neuroendocrine tumors is peptide receptor radiotherapy. So one thing that's also been considered for neuroendocrine tumors is whether we can add immune therapy to peptide receptor radiotherapy. And I've been working on this proposal. Again, here's a cancer cell, here's your somatostatin receptor, well-differentiated neuroendocrine tumor. And you know how Lutathera works. Lutathera binds here. And there's some idea that when you're giving a patient Lutathera, you're probably stimulating the immune system. Isn't that the perfect time to give somebody immune therapy? It's in order kind of like a 1-2 punch in order to get at the cancer. And as far as immunogenicity goes, meaning we talked about, when did, tumors start to look funny? So, one of the ways to make tumors look funny is to radiate them. And we've shown that in the lab, that if you radiate a tumor, they start to become more immune friendly. Your immune system starts to say, hey, something is wrong here. You get PD-L1 expression, which is a marker for the immune system. So that's another reason why the combination is promising.
Are you saying that increases the visibility of the tumor?
Right. So the study I'm proposing is to do the combination together. We've been trying to do it through the SWOG cooperative group. And a cooperative group is basically a consortium of cancer centers that's sponsored by the federal government. So I've been trying really hard to put this study together. And so far the feedback I've been getting is that a little bit concerned that maybe with the first time someone's getting PRRT is not the best time to do it. Maybe we should do it the second time they're getting PRRT. So the concept is being developed, and it looks like it's probably going to be an idea where if you're getting PRRT for the second time, we're going to try immune therapy with it. And here's the CheckMate study again. This was a promising early data. And this is what it looked like in CheckMate 032. This is for high-grade tumors now. So let's talk about the data for high-grade tumors. So immune therapy is approved for high-grade tumors. You can use it. And CheckMate 032, this is for small cell lung cancer, which is a high-grade neuroendocrine tumor of the lung. Here, one immune therapy was compared to 2 immune therapies. And what's always easier to look at these graphs here. And here you see these 2 graphs. The combination immune therapy did better than the single immune therapy. So this is why some of you may or may not have heard, but a lot of times if you have a high-grade neuroendocrine tumor, your doctors will recommend 2 checkpoint inhibitors together versus 1 based on this experience in small cell lung cancer. There's been another study that's close to accrual called the DART study. The DART study was part of the MATCH trial. And what they did is, and this was a big national study, patients who had high-grade neuroendocrine tumors were enrolled, and they were also given doublet immune therapy. And that data should be published soon. So to go back over everything, I would say the status of immune therapy now is that there's a very little bit of activity in the well-differentiated tumors, anywhere between 4 to 6% benefit in well-differentiated tumors when you don't select any one particular. Whether we'll be able to select that 4% out is to be seen. So for well-differentiated low-grade tumors, immune therapy by itself has not becoming a standard of care because we all don't think 5% is enough, right? That's not good enough. For high-grade tumors, though, immune therapy's already arrived, and we're doing it in the clinic. So that's pretty cool. All right. And one thing I also want to tell you about, there is a new clinical trial. A lot of patients have been asking me about it, so I figured I'd present it here today. This is what's called, it's a Zen Core Antibody. This is a bispecific antibody. And what this bispecific antibody does is it has one arm that binds to the T cell and one arm that binds to the serotonin, the somatostatin receptor on the neuroendocrine tumor. Okay? And by that way, it's able to kind of bring the immune system to the tumor. You see this? You see a T cell? and you see the tumor cell. And what tumors have SSTR2? Neuroendocrine tumors, right? So it's kind of like, think of it like this way, like one hand is holding the tumor, one hand is holding the T cell, bringing them together. So bispecific antibodies have been actually quite active right now in colon cancer. And there's one drug in particular that's moving through development. So these can work, so that's what's exciting. The only problem with them is that, can anyone guess what the problem might be with doing something like this? So when you mess with the immune system, you can get immune system side effects. So that's one of the problems. So they can be a little bit, sometimes you can have a little bit of an immune reaction to it. So for this study, we're keeping a very close eye on patients and giving the treatment as an inpatient in the hospital. I think that was the first tumor they started. So the bispecific antibody is a concept. So what you need is, so you need two things. This part is easy, the CD3 part. Because that's your T cell. Your T cell, that's fine. It's this arm that's hard. So you need to find a tumor that has a very consistent receptor expression of a certain receptor that you can use. So for neuroendocrine tumors, it works really well because a lot of them have the SSTR2, right? And they're pretty, you can do a gallium scan, you see all the SSTR2, right? Very few tumors have a similar situation. For colon cancer, they have CEA, which is another antibody. So what you can do for them is you can take a piece of the tumor, look at it under the microscope, make sure there's enough CEA, and then they would be included for that drug. So that's the problem, yeah.
Is immunotherapy an option if you have an autoimmune disorder?
That's a great question. It would depend on the autoimmune disorder and all the other issues. I wouldn't say it's an exclusion per se, 100%, but it depends on what's going on, how serious the autoimmune condition is, but it's definitely something to be concerned about. So, right, so immune system reactions, for the standard treatments that are approved right now, it's usually like an itchy skin rash. It's a cough because the immune system is causing inflammation in the lung. It can be diarrhea because the immune system is causing the diarrhea. So we call them immune-mediated side effects. For these new treatments, it's sometimes because they work in a different way, sometimes you can have a systemic immune response. They call it cytokine storm. It means a lot of inflammation in the body. Or cytokine release syndrome. Attack healthy cells. So the immune system, if your immune system is overstimulated, it can affect healthy tissue. Right. But an easy way to put a brake on the immune system is to give steroids. So in the clinic, they tend to be very safe medications because if you're having an immune side effect, we're able to stop it with either just stopping the medicine or give you a steroid, which immediately stops the side effect. The only hard part was in the beginning when we didn't know what was happening, and then that's when you get kind of in trouble because you don't know what's happening. But now so many patients have received immune therapy that.
Is it just shrinking the tumors or is it getting rid of them altogether?
Oh, so the question was, is this shrinking the tumor or getting rid of it altogether? Most of the time it's shrinking it, and once in a while it's getting rid of it altogether. Yeah, exactly. Remember those curves in the beginning I showed you? So yeah, that's what's neat about immune therapy. Sometimes it can get rid of all the cancer cells. Yeah. Well, all right. I hope I didn't talk too long. Thank you for your attention. All right.
Hello, everyone. I'm Jun Gong, new to the practice with Dr. Hendifar, but also seeing some neuroendocrine patients, and some of you I recognize in the audience. Thanks to the organizing committee, Dr. Hendifar, Dr. Thomson, for inviting me to give this talk on precision medicine in neuroendocrine tumors. It's a pretty broad topic, but an ever increasingly popular one. So to begin, I'd like to talk about the human genome. Do you all know what a gene is? It's essentially a segment of DNA that encodes for a lot of different proteins that help a lot with our biologic function. And as this diagram shows, our DNA that contains the genes are condensed into larger structures called chromosomes. Chromosomes are contained within the nucleus of the cell, and the cells are essentially our basic building blocks of what makes us who we are. Our tissues and organs, and as Dr. Hendifar mentioned, cells can also go haywire and become cancerous as well. So this is important to know because you may have heard of the Human Genome Sequencing Project. So the genome is the constellation of all the genes that make us who we are. And the initial draft of the human genome finished in 2001. It was an extensive effort, global effort, took about a decade to do, cost about $2.7 billion. And at the end of this all, they had mapped out about 3 billion DNA bases. This is why it took such a long effort. But at the end of this, we had our first draft of a human genome, a reference genome, because in order to figure out what's wrong, you must know what's normal. So why did it take so long? Why did it take 10 years, so much money? Because they were using essentially an old school kind of technology, Sanger sequencing. And what this means is that what they use is, so DNA, the way it's designed, there's 2 strands, double strand, and then double helix, right? If you know the identity of one strand, you automatically know the other side because they're complementary. That's the way it works. So they use a small segment of DNA called a primer, and that attaches to the target DNA of which you want to know, which is the template here. What you do then is you add a bunch of other single DNA bases, and they've designed it where they have a tag at the end. And what it does is when it binds to that complementary part on the unknown DNA, it produces a signal, a light signal. And this signal gets sent to a complicated array of computers that maps it out and reads out the sequence for you and reports it. The problem is this was being done single base by base. It was automated, but as you can imagine, 3 billion bases, doing it one at a time, even automated, would take a huge global effort. And that's the reason, that's the initial foray of how they sequence the human genome. Now, this is a timeline of the most important hallmarks in genomic sequencing. With the advances in technology, what they essentially did was, with the Sanger sequencing, they essentially developed technology that allowed it to do it massively parallel in sequence, just many Sanger sequencings at once. And these devices are known as next generation sequencers because they were the next generation after the original Sanger sequencers. Another terms that you may hear in the literature or in the news, massive parallel sequencing, high throughput functionality. These are all concepts that we're just doing it at a much more massive rate and capacity. The first of these sequencers was the Roche 454. That was, I'm sorry, it's a little blurry is it me or is that, yeah. It's, the first sequencer was released in 2004. This was followed by the Illumina sequencer and solid sequencers. These are just commercial names for the types of machines that they were using. And then the actual draft of the human genome finished in 2003. And with this reference genome in hand, they were able to now completely sequence an individual person's genome, which still costs about $14 to $25 million by 2006. So made some progress with some time, cost was substantially cut down, but still not quite there for commercial use. And with these recent technologies, we realized the genome has about 3,000 billion DNA bases. Not all of them are going to be implicated in cancer. Actually, a lot of genes in our genome don't have any function at all. So what happened was, with this reference genome and with these technologies in place, there were groups of government-funded agencies and also non-government funded agencies, privately funded agencies, such as the ENCODE, Cosmic Initiative, ICGC, TSP, TCGA, and they all had a unanimous goal to systematically catalog all the mutations that have been implicated in adult and pediatric cancers. So now we're moving beyond the human genome and focusing more on cancer genome. And this is very important because this is essentially what the field is leading now into for using precision-based therapies in cancers and such as neuroendocrine tumors. Before we kind of talk a little bit more about what we've done in terms of precision medicine in neuroendocrine tumors, next generation sequencing technology broadly can be characterized into two types. One is a targeted panel of recognized cancer-associated genes. This means investigators have said in neuroendocrine tumors, based on the research we have, we only know a certain amount of genes cause cancer or are associated with cancer. So we want to narrow this list and produce sequencing of this set list of genes. The other approach is to do more of a shotgun approach. It's to kind of panel the entire genome or the entire exome. This is called whole exome or whole genome sequencing. This is more of a shotgun approach, scans a lot of cancer genes that we know of and searches for it within each patient's tumor tissue. Now, as you can imagine, number two, because of the more wider grasp of reach that it's testing for, in terms of costs, it's relatively more costly than a targeted panel. But nonetheless, we have now reached an age where next generation sequencing is very commercialized, and they have gradually made its way into routine clinical decision making. We certainly still use it for research purposes, and we still use it for discovery of novel disease-causing alterations as well. This is research I did a couple years ago. I essentially reviewed a lot of the commercially available next generation sequencing companies that are available. A lot of the common ones that you'll see are Foundation One, Caris, Tempus is one that Dr. Hendifar and I have referred to as well. And in terms of sample requirements, most will require tumor tissues that have been preserved by your pathologist and stored in the hospital in what we call FFPE, or fresh formalin fixed paraffin embedded tissues. This is just the preservative that they put the tissues in. It looks like little blocks of plastic essentially. And that's kind of the standard tissue type that these next generation sequencing companies require. In terms of the actual sequencers that they're using, they use the most commonly commercially available ones like the Illumina and the ion torrents that we discussed before. And now this is the third column here, or the fourth column here is the genes covered. This is it's a commercial industry. So next generation sequencing companies will say, hey, my test here covers 315 genes. How much does yours cover? Other companies, for example, Foundation One covers 315, Caris over 600, Tempus is around 1,000, actually. And this is, one way of, it's a commercial industry, their pitch, essentially. For us as clinicians, we know that there are a finite number that have targeted therapies available. So oftentimes, yes, it's good to know more information, but oftentimes we're left with a lot more information of uncertainty, what we're doing with these other mutations that they're testing for, but yet there really isn't any targeted therapies available, nor has it been studied before. These are all certainly hypothesis generating in that they can lead the next studies about figuring out what these mutations offer. And then cost is always a concern, right? So here I've listed out the costs. As you can see, we've made a lot of segue from the $1 billion genome to now a few $1,000s per each individual patient. And then time of turnaround is a very important question that patients always ask. How long are these next generation reports going to come back? And here a range about 14 days. Some say they can even get it to you within the first week. In our experience, it's about a few weeks, 1 to 2 to 3 weeks turnaround time. And then there are also some hereditary cancer genetic testings that are also available as well commercially. So now let's talk about our research that Dr. Hendifar and I have done in terms of looking into neuroendocrine tumors. What we did was collaborated with a company called Perthera. This is a unique company in that they're not actually performing the next generation sequencing. What we do is we send out the tissues to them. They send it out to the next generation sequencing companies, and then they condense it, filter out what's important and not, and provide a consensus expert report of your mutations and what targeted therapies and what clinical trials are available. And what we did was we investigated a cohort of 46 patients. As you can see here, the median age was 52 years. The majority were female, 52%. The majority were stage 4 pancreatic neuroendocrine tumors, 54%. These are metastatic, meaning the cancer has spread from within the organ from which it came. The majority, 65%, were low intermediate grade, and the most common site of biopsy was the liver, followed by the pancreas. This meaning the most common site of where the tissue came from, for which we did the next generation sequencing. And what did we find? So here the top half is the genomic sequencing, and the bottom half are actually the proteins. So this is genomics, and then the bottom is proteomics, meaning the protein expression levels of these patients' tumors. And as you can see here, the most common 2 mutations that we found in pancreatic neuroendocrine tumors were those of MEN1 and DAX. You can see the percentages in the third column here. And we also listed in the middle column what role do they have biologically as a gene. These were involved with remodeling your chromosomes in which your genes are contained with. Another pathway that was commonly altered was replicative stress. Mutations in TP53. This is a gene that is involved in regulating your cell division. So as you can imagine, if this gene is mutated, there's not an off switch. Cells continue to grow and replicate. And there were certainly several mutations that were common in Mtor signaling. And this is not a surprise. There's actually an FDA-approved Mtor inhibitor used in the treatment of metastatic neuroendocrine tumors, everolimus. That's an Mtor inhibitor. And this is, again, providing rationale that this pathway is altered and why this drug may work and is effective and has been established. In looking at the proteomics, the proteins, again, Mtor signaling was the most commonly altered. I wanted to highlight here too also, however, we found a couple of proteins that were altered that actually have been implicated in resistance to chemotherapy, not so much in neuroendocrine tumors, but in other cancer types that have been explored. And Dr. Hendifar also mentioned earlier about immunotherapy. We also found some proteins expressed that, they were somewhat there, 20% for PD-1, but not as high as we'd like in other tumors to be a very good response to immunotherapy. And the 100% for microsatellite status, this is actually, we would require the opposite, MMD instead of MMR to predict for response to immunotherapy. Instead here, 100% of our patients were MMR, meaning that they had a low amount of those mutations or disfigurements, as Dr. Hendifar said, that would allow your immune system to identify this type of tumor. Now, what we did was we then condensed all of these alterations and put it into a heat map. And what this does is it allows us to kind of visualize if there are certain patterns that we're noticing with mutations and clinical characteristics. Down here are all the patients that have been identified, all 46. The column on the left here are all the mutations. The row on the top are things like gender, age, diabetes status, smoking status, advanced stage, and high grade. And they either have a yes or for Y, meaning they have this present, or N for no, F for female, M for male. And I've kind of highlighted the first kind of pattern that we notice. You can see here, these mutations in TP53 and RB1, they seem to have been clustering in all those patients that have yeses for high grade. And this, why I'll actually show you a little later, that yes, this was in fact true. A lot of patients with these mutations did have higher grade tumors. And then another cluster that we notice, as you can see here, mutations in MEN1 and DAX, as you can see, they tend to occur in patients that did not have advanced stage disease or those that did not have high-grade disease, meaning MEN1 and DAX mutations were more commonly found in patients with lower-grade tumors or non-metastatic disease. And so this is actually kind of a summary of the relationships that we found. And as I mentioned, RB1 and TP53 mutations were more likely associated with high-grade tumors. On the contrary, mutations in NGS and actually being of age greater than 54 meant that you had a lower likelihood of having metastatic disease at diagnosis or a high-grade tumor. And then what we looked at is we tracked the outcomes, the survival of this cohort of patients. And we wanted to see if there were any mutations that when you were detected on your tumor would predict how long you would live or how long your survival was. And as you can see here, there was only one pathway that was significant based on our statisticians' analysis. Here, if you had an absence, which means you did not have this type of mutation in the replicative stress pathway, the most common ones were TP53 and RB1, that you were put in that top blue bar there. And look at that blue bar. They're still going 10 years out, right? And then if you had these mutations, TP53, RB1, they didn't do so well. Their outcomes, as you can see here, at that red curve right there. So what does this all mean? This is essentially what the field is going. This is what precision medicine means. You profile your tumor at some course along your therapy. Most patients may not get their tumors profiled at the beginning, but at some course of their treatment. And we identify potential mutations that are actionable. Whether these means that we put you on a trial for a targeted therapy, or we have targeted therapies approved for this availability, or they serve a prognostic implication. They tell you how aggressive your tumor is, how likely you are to do well with your tumor. And this was an initial study that was very seminal. It was published in Nature. And so how did it pan out compared to ours? Well, if you look at here, men 1, 41%, almost identical. DAX here, 22 and 28%, fairly comparable. ATRAX, 10 to 7%. But in one of the Mtor signaling pathways, P10, 7 to 18%, which still meant that there was some differences in our cohort of patients compared to the cohort in the Nature study. And I'm almost finished. I didn't want to bias. Just because we did pancreatic neuroendocrine tumors does not mean there's not active research going on in a lot of different tumors. So there's papers coming out in profiling in pulmonary carcinoids. There's papers coming out in small bowel or mid-gut neuroendocrine tumors as well. There are molecular profiling in a broad array of neuroendocrine tumors. So it's a very exciting time in cancer genomics. And lastly, I've been being asked a lot more about this. You may be seeing this as well. Circulating tumor DNA or circulating cell-free DNA. This is a unique and novel approach to genomic sequencing. Instead of doing it in the tissues, you're doing it in the blood. So this is still, it hasn't hit as mainstream as in other tumors, for example, lung cancer, but there has been pilot studies. As you can see, there were only 10 patients done in this cohort here. And what they saw was that tumor DNA was actually detectable in the blood of patients with neuroendocrine cancers. But however, M1 meaning metastatic. So they were only able to detect those black bars there. Those are the tumor DNA that they're picking up. It was only being detected in those who were metastatic, not so much in the non-metastatic, which is all the gray bars on the left. And theoretically, it makes sense. If you are metastatic, you are likely to more have tumor-shedding cells that are able to be picked up in the blood, rather when you have a localized tumor. So for every good pilot study, their first priority is to show, is this test just as valid as we're detecting from our tumor tissues? And they did show this. They found out that alterations detected in the blood showed good correlation with the original tumor tissues. But they also found out, and this is, I think, what a unique twist of what circulating tumor DNA offers, is that this patient, they reported him or her twice, the number 7. This one, they actually showed that over time, as the tumor grew, you were able to detect more tumor DNA in the bloodstream. So this kind of presents a novel idea that, for example, you could follow a patient serially over time with circulating tumor DNA to not only find out what mutations that they have that may guide therapy, but also an idea of what your tumor burden and what your tumor is doing. And as you can all imagine, this is much more easier to draw blood serially than to have a patient undergo repeated biopsies over time for the same purpose. So this is one area of interest that I wanted to kind of introduce the concept to. So then finally, where are we right now with precision medicine? We've certainly made a huge progress from the human genome age that took 10 years to profile a tumor to now where you can get an individual's tumor profiled in a matter of weeks and more cost effective as well. It still needs further validation. You know, our cohort here was only 46 patients. We need a larger sample size of patients to make sure that our findings are reflective of the general population. And these are needed to find, again, what precision medicine means, markers that have prognostic value and predictive value, meaning predicting that you'll have benefit or not to a certain therapy. Because if you're not going to have therapy, we have plenty of other options that we need you to get on in order to control your tumor. We don't want to be wasting time with a chemo or a systemic therapy that's not going to work. Larger studies, ideally, if it's going to be an investigational, clinical trials are the best type of things that we often recommend our patients. And here's just a sampling of all the national clinical trials that are doing matching. They're profiling your tumors at diagnosis, and they're matching it to targeted therapies that are available. And this is, along with our research and research going on with a lot of groups in the country, we think that precision medicine, our goal is to make it a routine clinical practice in the near future. Okay. Any questions?
Thank you very much for giving us the full. That was great to take us all the way from the beginning. So we have some questions that came in ahead of time by email. And also I wanted to ask you, are any of these now covered by insurance to have this testing?
These companies are very equipped and they've streamlined it into their processing. Financially, there shouldn't be an issue, regarding that.
Okay, and that's available at Cedars, or will?
Yes, so
Okay.
Dr. Hendifar and I send out our, like I said, at some course, we do certainly send out next generation sequencing, and we're hoping to do this on all of our patients.
Excellent.
Yep.
And in general, do the carcinoid types of neuroendocrine tend to have a lower mutation rate than other types like PNETs?
So Dr. Hendifar kind of showed that slide earlier of the mutational burdens, and certainly compared to, melanoma, which was that big bar of mutational burden, carcinoid neuroendocrine are more towards the other end of it. The four highest mutational burden tumors, melanoma, lung, bladder, But there are ongoing strategies, as Dr. Hendifar mentioned, of overcoming that and making our immune system able to react and target even tumors that are thought not to be as immunoreactive compared to melanoma.
Great. And if you could, one of the patients emailed this question. If you had an SDHX patient, and I'm not sure what that is, but maybe you can help us, in your office, would you recommend a TK1?
So I think SDH they're talking about succinate dehydrogenase, and TK1, do they mean TKI, meaning perhaps
It could be
tyrosine
TKI.
kinase inhibitor?
Yes.
So that's an example of the reports that were generated from these next generation sequences. What they do is these companies, and they do have scientists, clinicians involved in their board panel as well. What they'll do is they'll peruse the literature for any kind of study that has shown, for example, SDH. They may find a study that was done in, bladder cancer, and they treated this patient, or they treated mice, for example, or rats with a TKI, and that happens to be, they weigh the evidence, and then they make a recommendation. So that's a positive, that's a plus, that it offers a potential for targeted therapy, but this is always an important decision from the treating oncologist and the patient regarding what options are there that have been proven, or what options that are certainly experimental and worth discussing. So it's a very very complex picture, but I believe it needs to be discussed with the treating oncologist team and the patient, both ways.
This is why we always talk about a multidisciplinary team for our tumors. So thank you so much, Dr. Gong. Dr. Thomson's working in nuclear medicine here at Cedars-Sinai and going to talk with us about PRRT. Welcome, thank you.
I'm talking about PRRT, which is the abbreviation for Peptide Receptor Radionuclide Therapy. It's a molecular-based approach or precision medicine, an example of targeting a tumor based on its surface receptor. The somatostatin receptor that's expressed in these tumors is targeted directly for the treatment delivery. And I like to think of it as a Trojan horse because you're sneaking the treatment into the body and then delivering it where you want it to work. The Trojan horse packed with cancer-killing radiation. How do we deliver the radiation with PRRT? The protein that binds to the somatostatin receptor is connected into a radioactive material. The treatment is given then through the blood system, and it circulates to then bind to the somatostatin receptors being expressed at sites of tumor. That therapy is then internalized into the alive tumor cell and trapped there where it delivers the energy that you've attached to that molecule. The energy is called a beta emission. It goes a very, very short distance in tissue and delivers its energy right there at the site of the tissue. And so you minimize the damage to the rest of the body in targeting just the surface receptors on the tumor tissue. What all treatment aims to do is to not only increase survival but also improve the quality of life and reduce symptoms and hopefully prolong overall survival. But you should see with that a reduction of tumor size if the tumor is shrinking back and being killed off by the radiation in the same way that you would if the tumor is being killed by externally delivered radiation or other chemotherapies. The image there on the bottom right is a whole body image for a patient with metastatic neuroendocrine tumor that is expressing somatostatin receptors, and the image is created from a gallium-68 scan, which is the net spot scan. The distribution of the gallium-68 dotatate predicts for you where the radiation is going to be delivered when you give the lutetium Lutathera PRRT. And this is the interface between diagnostic imaging and then delivering therapy. And someone's come up with the terminology theranostics, which just gives us another word to debate how to spell it and how to say it. But theranostics is that combination between targeted imaging and then delivering a radioactive material to deliver therapy in the same distribution as the tumor. These little black hotspots, they look larger than they are in reality because of how bright the image is with the little tracer stuck to the surface receptors, but it shows us, compared to the background, how that tracer is really intensely accumulated by little sites of disease, and that will predict for you where the therapy is going to be delivered to. So if we take a step back and say, what about the non-midgut, the non-pancreatic tumors that are neuroendocrine and express somatostatin receptors? They are the minority, and in this diagram from a really nice article in 2017, bronchopulmonary here was displayed as around 30% of the spectrum of different origins of neuroendocrine tumors from the gastrointestinal tract and the pancreas. Of the bronchopulmonary neuroendocrine tumors, the majority will express and have a surface receptor, the somatostatin receptor, that can be targeted for imaging and treatment. In reading all about this, I found myself in a whole world of cancer and history of how tumor types are defined and World Health Organization categorization that's evolved over time. And I found just hours and hours and hours of confusion as a physician. It's very complicated, and it's certainly not my traditional comfort zone, which was cardiology and then general nuclear medicine, but trying to get my head around it as well in terms of which type of lung neuroendocrine tumors are we talking about. What we're talking about when we're talking about delivering PRRT to non-midgut tumors, we're really talking about the types of neuroendocrine tumors of lung origin that are metastatic or will progress and behave in a cancer kind of a way, but the ones that tend to spread and grow slowly are the ones that tend to have the somatostatin receptor expression that allows consideration of PRRT. And the neuroendocrine origin tumors in the lung that spread more aggressively and more rapidly are not the type of lung tumor that I'm talking about as a PRRT therapy approach. Because when you deliver PRRT, as we'll get to towards the end here, it takes many months to deliver the full course of treatment. So you really need to have a disease that's got targets and is also moving at a pace that allows you to deliver the treatment and get the response you want. There are publications that have reported on the use of PRRT for people with lung origin neuroendocrine malignancy. And this is way too complicated, so we'll just take out some of the words. The studies that I was reading about date really predominantly from the last 3 years, and predominantly European, with the availability of PRRT in Europe that predates our market availability of Lutathera by a decade. There's really much more long-term experience and more breadth of experience in Europe with delivering this kind of radioactive therapy. And as you can see there from the numbers, really it's a very small total number of people with bronchial or bronchopulmonary origin neuroendocrine tumors who are included in these bigger studies of delivering PRRT to neuroendocrine malignancies. But it does give us some sense of how that type of tumor will behave in response to treatment and whether the treatment should be an option for someone who has a neuroendocrine tumor that's a lung primary. And really the numbers are similar to the progression-free survival rates and overall survival rates, either disease stability, partial response. The numbers are quite similar, maybe slightly less, but in the same ballpark as what you see with the midgut and pancreatic tumors. And I wanted to just focus in on the one study that's the European longer-term data for 443 patients who've had a cumulative dose of the radioactive material, to point out that there were a group of that study who had the bronchial origin neuroendocrine tumor. The overall progression-free survival in this study was 29 months. Oh, don't do that, I might delete something. And overall survival 63 months with quite large ranges. But what's the point of this? Well, the point is this is one of the bigger, longer-term studies of people receiving this kind of treatment. It gives us some information about how effective the treatment was. It tells us a little bit about what the long-term toxicity profile is of delivering a radioactive medication, because the last thing I want to do is make someone's situation worse. The whole point of being in the healthcare business is to make someone's situation better and to avoid any unforeseen side effects, ineffective treatment, or just radiation exposure for nothing. We have some concern about getting radiation exposure to our bodies when we think about what our chances of developing a cancer. The rate of leukemic type events or myelodysplastic bone marrow changes in people receiving radioactive material for treatment of metastatic neuroendocrine tumor is down under 2% for the population for this largest study that's looking at longer-term outcomes. And importantly, within this cohort, who are selected as being ideal candidates, there was nobody with long-term therapy-related kidney or liver failure problems. So really the toxicity profile is pretty good for these treatments in the longer term. Then if you look particularly at the non-midgut, non-pancreatic subset within the study, that's only 23 patients. When you look at who's in the study, it gives you a sense of how the study data might be then applied to a broader population. It's a mix of people with different intensities of expression of the somatostatin receptor. And that's interesting to me because my brain wants to know how hot or active does a gallium-68 scan need to look before I would say, oh yes, this person's going to get a benefit from radioactive material delivered and targeting those receptors. The majority of people included in these studies have really quite intense expression of the somatostatin receptors. So when I'm looking at a PET scan, for instance, and grading the intensity of the tracer at the sites of disease, I want to get a sense of whether it's really very obvious or maybe only just mildly obvious compared to the background organ tissue. And that will predict for me whether or not someone's likely to get a benefit from this kind of treatment. They tend to be well-differentiated tumors, meaning they're organized in their structure, they are closer resembling to normal tissue, slower growing and expressing a lot of surface receptors. These are the people who have responded, who have been part of this longer-term study. So knowing things about the tumor that are not necessarily imaging-based, but can be imaging-based, the pathologist can tell us about the growth rate. When they talk about mitotic index and Ki-67, they're trying to express to us how rapidly that tissue is turning over and growing. And you can get signs of that just looking under a microscope at the tissue samples. And as you've heard from Dr. Gong, I'm sorry I didn't understand half of what you said, but it sounded really good. You're probably thinking the same about my talk, yeah. The growth rate and the way in which tissues turn over and grow and tumors behave, it can be all determined from a much higher level of who we are genetically and in our DNA. And it can also change over time in tumors as somebody lives with their tumor for years and years and years. The tumor behavior can be slightly different over time. So re-looking under the microscope can be important in trying to work out whether you've got a tumor that's a slower growing behavior or a more aggressive behavior. And then we use a radioactive glucose for our PET scans. That tends to tell you about just general growth rate. Things that are growing are hot on a glucose PET. Things that have somatostatin receptor expression that may be growing very slowly will not be seen on an Fdg-Pet, but will shine bright on a gallium-68 PET. And in some situations we use both of those imaging approaches to get a sense of the behavior of the person's tumor so that we can then decide whether or not the treatment's likely to be beneficial, to offer some improvement in symptoms and ideally overall life expectancy. All right, that's a lot on the concept of lung and tumors and targeting receptors, but this is just a side comment about other tumors that express somatostatin receptors. I think these are zebras as well. Are we allowed to call them zebras if they're not carcinoid? But neuroendocrine type tumors that secrete materials can also have somatostatin receptor expression, and we've been sent patients to consider for PRRT who have metastatic paragangliomas that are also rare, can also be related to genetics and mixed into other syndromes, but they tend to have overexpression of somatostatin receptors. And this is another potential application for the targeted treatments as we learn more about how to personalize medicine depending on the individual's tumor behavior. There are two different options for therapy potentially with someone with a metastatic paraganglioma, and that's related to the type of surface receptors. The commercially available radioactive iodine, MIBG, is now approved for use in the United States. We are not giving that at Cedars yet because it seems to me quite a lot more complicated than safely delivering the lutetium Lutathera. So for us to be able to do that, we would need to have more software, different physicist support staff for dosimetry calculations, and specific facilities for inpatient treatments, possibly. But for this particular tumor type, that targeted treatment will give symptomatic relief for 75% of patients living with metastatic disease. Using Lutathera for something like a metastatic paraganglioma is officially, it's an off-label use because that's not what the FDA has approved the drug for. We have very limited experience with it. We have 2 patients referred with metastatic paraganglioma, one of whom is receiving Lutathera. And the published data of the use of Lutathera for this type of tumor is retrospective and quite limited, but it will come as we start using these agents more. So who gets referred for PRRT, this targeted radioactive material? It needs to be someone who has a somatostatin receptor expressing disease. Oh, am I out of time? Nope, okay. And it needs to be someone in whom the basic organ functions are reasonable in the background, because even though the treatment's targeted, some of the rest of the body is going to see the radiation as it's circulating through the bloodstream, and excess is cleared through the kidneys within the first few hours. So we have some parameters there in terms of basic kidney function, basic bone marrow function and liver function that need to be at a level of sufficient health to be able to safely give this kind of treatment. This one down the bottom here sounds a bit weird, doesn't it? Inability to comply with radiation safety requirements. It's because the radioactive material is in the body and excess is cleared out through body fluids, especially the urine. Anyone who's had this treatment, who's going out into the community, needs to be sufficiently able to care for their bodily functions or have assistance for bodily functions that doesn't endanger anyone else in the house, because we have a mandate not to expose someone else to radiation who didn't ask for it and doesn't need it. Especially if you can imagine the scenario of someone living in a household with toddlers or a pregnant woman, and what the potential lifetime impact of radiation exposure might be to somebody who's very small, who gets an unnecessary exposure to radiation. Having said that, for me to get irradiated by treating a patient when I'm in the same room, I've been told by the radiation safety staff that I'd actually have to have chest-to-chest hugging going on for about 2 hours for me to get the equivalent of a couple of x-rays. So we're talking about a tracer that goes locally and stays locally and doesn't emit a whole lot of energy. People who have this are not like shiny light bulbs giving radiation off. It's all internal. But their body fluids could potentially contaminate areas around the house. So that's one of the factors that we talk about a lot in planning treatment. We get right into the personals of people's toileting habits and continence issues and who handles what, and then right into very detailed planning for someone who does have urinary incontinence, who might use pads or materials that they would ordinarily toss in the garbage. If they're coming for Lutathera, they have to buy a plastic bucket with a lid and plan to store all of that waste for a period of several weeks before it goes out in the main garbage into the community processing facilities. That sounds science fiction, doesn't it? But it's our community processing facilities have radiation detection equipment in them. They're there designed to detect someone trying to poison our environment. So even medical radiation, we need to contain it and not put it into a community waste disposal area until it has decayed, meaning its energy has dropped away to a background level that's not going to set off alarms. And the implication for our facility with its radiation license is that if we were found by the state to be poisoning our environment or endangering members of the public, then our license could be revoked, and that would be the end of X-rays here at Cedars-Sinai. That would be bad. I don't want to be the person who's responsible for that. How do we give it? We have a day-stay procedure center that's been set up. We give it as 4 doses at 8-week intervals, and each dose is delivered intravenously over 30 minutes. There is an amino acid infusion that is run for several hours, and that protects the kidneys from damage as a bystander organ. And the amino acid infusion itself can cause quite profound nausea, so we need to give antiemetic support during each visit. That's a fancy word for trying to not have our patients feel ghastly and throw up, so I want them to feel as comfortable and relaxed as possible. And then at the end of each treatment visit, the long-acting octreotide is given. It's by prescribing guidelines to be given within 4 to 24 hours after each treatment. We choose to give it here because so many of our patients are coming from miles away or even from out of state, and I want to know that they've got that in their systems before they leave. This is what our little center looks like. We currently have quite a large room with 1 bed, although it does have the capacity for a second bed to be in there during a treatment day. We have a dedicated infusion pump, and the stuff is administered behind a Perspex screen. Just a thick layer of plastic will stop the energy from going very far. There's a TV on the wall. That's important because that's one of the chief distractions during the day. And the process for getting the treatment starts with a referral from the oncologist. Generally, it's most often been Dr. Hendifar, although I have had some contact with other oncologists directly from the Los Angeles clinic or from out of LA. The first step would be to review all of the clinical data and the image data and to discuss the situation at our multidisciplinary conference so that we are all in agreement that that's a reasonable treatment option and that there isn't any better idea, like, for instance, a targeted liver treatment if the disease is in one place in the liver. Having assessed from records that somebody looks like a candidate for this treatment, we would ask them to come and visit and meet us, look at the place, meet the staff, meet the doctor, meet the radiation safety officer, and hear about the treatment, as well as giving us an opportunity to reassure ourselves that the person is going to follow instructions for radiation safety and looks healthy enough to receive the treatment safely. If someone's receiving Medicare for their health care, then we can go ahead and schedule. We now know that Medicare is covering this treatment. If you'd asked me 8 months ago, I had no idea, but now I do know that Medicare is covering this treatment. If somebody's privately insured, then it's a little bit more of a complicated setup where the company making the stuff actually interacts with the person's insurance company and with the patient to put in place a contract that will cover any shortfall of the insurance pre-authorization. I think that's how it works, although I'm definitely not an expert when it comes to private insurance. This side of things for people who are not receiving Medicare can be quite long and drawn out. Once we've got through that process, then we can go ahead and schedule the treatment and see someone 4 times at 8-week intervals. Now, this is HIPAA compliant because this gentleman, who shall remain nameless, has given permission for us to use his image in our materials advertising our program, and he's our first official completed treatment patient. And it was certainly a joy to get to know him and his family over the multiple visits. We gave our first treatment at the end of April. We've had 33 patients referred to us over the last 12 months, thereabouts. We have now 5 people who've completed the full course of treatment and 18 people who are currently receiving treatment. Our appointments on the calendar go right through to September. 6 people still in the process of trying to get their insurance and funding sorted out, and 3 referrals who've gone ahead to have some chemotherapy first with our PRRT on hold but in the background. Most recently and most exciting for me is the Pharmacy and Therapeutic Committee here at the hospital approved the use of a 2 amino acid infusion product, and once they have chosen their site to buy the product from, we should be able to give a 2 amino acid infusion instead of the multi amino acid infusion, and that's relevant for comfort, because a 2 amino acid infusion should have much less nausea associated with it when it's given during the treatment visit. Who's getting treatment? This is just a little snapshot. Obviously, nowhere around the country has a very large experience of people referred for this type of treatment, since it's only been available to us commercially since the beginning of 2018. But our patients referred are widely varied in terms of age, tend to be more women than men. I think that's the type of population you see with this type of tumor, but someone will correct me, I'm sure. Most patients have had a gastrointestinal or pancreatic primary. 2 people referred with a paraganglioma, 2 people referred who've had a lung primary, 1 who has completed treatment who had a renal primary, and we have 2 people who are in process to receive treatment who've had treatment before, either in Europe or through a clinical trial many years ago. Second time around, PRRT is again evolving in terms of the world's knowledge base about how well it's tolerated, but it appears to be relatively well tolerated without much in the way of difference between the first and second time for bone marrow toxicity or risk of leukemia or myelodysplastic syndrome. But I counsel patients that the more radiation you have in your lifetime, theoretically, the greater your chance of triggering some other kind of event. So radiation, although we all live with it from the sun and from the ground, and life doesn't exist without it, we certainly believe that we don't want to have it unless we actually need it. So that was as far as I was going to go, except to say that the molecule there provided a little framework for all of the people involved, because like our multidisciplinary team discussions, just something as simple as starting a new service and delivering a new product has just extraordinary complexity and little octopus limbs. Every time you think, yes, we'll do it that way, there's another committee and another person involved, and it certainly has taken a lot of collaboration between nursing and pharmacy and our nuclear medicine people, our facilities management people, insurance, and the biggest one there, the patient. I think our patients are extraordinarily wonderful people, and they've been just a pleasure to meet and to be involved in the care. So I can answer some questions. Did I go on too long?
No, not at all. Thank you. We have a few questions that were submitted, and then maybe we can open it up to the room. One of them is, what is the role of combination therapy with chemotherapy and PRRT simultaneously? And would you use this? And if so, what chemo agents? And I realize that would be a question for Dr. Hendifar.
And Dr. Hendifar does have some thoughts on this. I'm sure he'll address them when he's back in the room. But from my point of view, the patients I've met who are taking everolimus tend to have bone marrow function that's already looking like they're taking something. So the hemoglobin and the platelets will be potentially slightly lower. I don't want to go and give another agent on top that's going to potentially impact the bone marrow further. And I'm shy of doing anything that's going to make somebody more unwell. There are clinical trials that are looking at the combination of chemo with PRRT. We could certainly explore being part of something like that moving forward, but in the first few months of establishing this program, my chief aim has been to not do any harm. And so I have not been treating anyone who's receiving chemo, and everolimus has been stopped a matter of a few weeks before the PRRT to make sure that we're not double hitting the bone marrow.
And that makes sense with some of what you presented about who's a good candidate. So, can you address bone metastases? What are their significance, and what would you do to treat them? Would you treat with PRRT for bone metastases alone?
And this is a really good question. And in a PET scan with the NET spot, when there's disease in the bone, it can really be quite alarming to see what that scan pattern can look like. If you consider that the spine, the pelvis, and the proximal portions of our humerus and femurs, that's where the bone marrow is. If somebody has an extraordinary amount of disease in and around their normal bone marrow, I'm more nervous about whether or not I will impact the bone marrow through killing off normal bone marrow cells. Having said that, people are individuals. If somebody's young and otherwise has a very vigorous bone marrow, then it's going to bounce back relatively quickly. And even if you do cause a transient dip in the hemoglobin and the platelets, it would be unusual with this treatment for it to be extreme toxicity. It would be unusual with this treatment to, say, have a situation like you sometimes get with the systemic chemotherapy, that someone's neutropenic and at risk for infection. We don't have that kind of impact on the bone marrow, but we certainly would take that into consideration. Most people do not have a picture of bones completely involved, almost every bone. Most people who have some osseous metastatic disease will have just little bits here and there. And certainly if there's only one region that has bone disease, then sometimes a targeted radiotherapy can be a more sensible approach than a systemic treatment. For instance, if it was just one lesion in one hip, then maybe delivering some radiation just there would be better than giving something like systemic treatment.
Great. And a lot of patients ask about SUV numbers. Can you speak about that, what they mean in terms of uptake, and also what you're looking for in the follow-up after someone completes 4 treatments of PRRT? Are you looking for those SUV numbers to go down?
And the SUV numbers in the nuclear medicine community, it's sometimes referred to as the silly uptake value, but it's a standardized value that's determined from the image, and it's a way of putting a number to the brightness of an area. And we know what number normal liver has, and then we can contrast normal tissue with tissue that is tumor with somatostatin receptors. So a normal liver SUV value might be something like 8 to 10 with our NET spot, whereas someone with a tumor that's expressing a lot of somatostatin receptors will have an SUV value that's 50, 60, 70, 100. So 8 to 10 times more intense than normal background liver. That's what we like to see, is something that's definitely more intense than background liver, ideally more intense than spleen. The uptake values, if you were to do the same test on the same person at 1 week intervals, you might get slightly different numbers. So it's not accurate to the decimal point. It depends on the busyness of the tumor on that day when you give the stuff. But they can give us a ballpark to track over time. And with a lot of cancers, we follow SUV values to prove response to treatment. So, for instance, someone with disease that is SUV at baseline of 50, and after a course of chemo, none of the lesions have an SUV more than 10, I would be able to say they're still active, they're not completely dead, but the amount of receptor expression has decreased, telling you that you've had a treatment effect.
And when are you monitoring people with follow-up gallium-68 scan after their PRRT?
This conversation comes up a lot because people want to know whether it's doing what it's meant to do. Did it work? And how do we find out whether it's working or not? I've said to patients coming for treatment that I think if they come into the course of treatment as a selected candidate, we should complete the course and then make a judgment about the effectiveness, and that I would not suggest they go back and have more restaging halfway through the treatment course unless something has changed clinically to give them a sense that something bad, the disease is progressing, it's not working. Yeah, but if they're otherwise feeling like things are the same or better, then we will give 4 treatments before someone goes back and has another MRI and gallium. And the restaging, the follow-up imaging, it should ideally match what was done at baseline so that you can compare apples to apples. So if your disease was characterized best by MRI, then you would choose to do the same kind of MRI at the follow-up so that you could look at the images side by side. And it may be that it's a combination of the gallium PET with the MRI, depending on where somebody's disease is.
Great. And you also mentioned that you had 2 patients who had had previous PRRT and now were having PRRT here. Do you know how insurance is covering that for someone who's had PRRT either overseas or through the right to try in the U.S.?
I don't know how that's going to pan out for private insurance, but and I don't know enough about the pre-authorization and insurance conversation to predict it either. It seems to me, though, that if you participated in something that was a clinical trial or you sought and paid for your care overseas, it should have no bearing at all on what you're insured to receive now that it's commercially available. Right. The bigger question to me is whether your body's in a state to tolerate it and whether it's likely to have a beneficial effect on the disease when it's given a second time.
There was also something in your slide about post-surgery or liver-directed therapy. Is there a certain time span that you recommend waiting?
So the support from the company has been really good for setting up to do this treatment, and the advice that I've had from the company and looking at how people were selected and enrolled in clinical trials and how places in Europe are handling things is it's the same concept as not doubling up with chemo and PRRT. If somebody's receiving radiotherapy, let's get that done and see how the body recovers and give it a little chance for the bone marrow to recover from that radiotherapy before we go and do another treatment. If you've just had surgery and a wound is healing, that's not the right time to be giving a whole body dose of anything radioactive. We need to just let the body recover a little bit.
Great. I think we covered most of the questions. There was one more. What is the role of Fdg-Pet scan for Gnats? And I know that you had that in your slide.
That's complementary to understand about the behavior of the tumor in terms of whether it's aggressively growing or not. So the gallium-68 will tell you about the somatostatin receptor expression on the tumor, and the Fdg-Pet will give you some information about how aggressively that tumor is turning over and growing, because things that grow take up more of the radioactive glucose. And indolent tumors and slow-growing disease tends to be PET negative or have low SUV values by Fdg-Pet. More aggressive, poorly differentiated, rapidly progressing disease tends to have higher SUV values. And putting the 2 images side by side can give you a sense of whether all of the sites of tumor in the body are behaving the same way or not, and then help you predict whether or not you're going to get a good response from the treatment.
Right, because we've learned that many of our tumors are heterogeneous, so they may not be the same Ki-67 when they biopsy different places or even biopsy the same tumor. Congratulations on the 2 amino acids. That's going to be.
Yeah, we're looking
is
forward
that
to that.
Actually implemented soon?
It is actually implemented soon. I can say it out loud. Yeah.
That's great.
The hospital is inspecting the facility. I think they're going to source it from somewhere in one of the southern states, but they need to be sure that where they're sourcing it from has adequate sterility and passed all their inspections, and that it's all about safety and not doing any harm in the process of
Well,
trying
this
to
is
do some
great
good.
news for us as patients to not have some of the nausea associated with the amino acids themselves, and also great for the staff here not having to deal with patients who are having nausea.
We hope that that will allow us to increase the number of appointments we have each week to offer as well. And it may potentially mean that we have 2 people come on the same day and stagger their visits. So obviously would be very careful to pair people up who would be okay being in the room together. But that's one way to improve our efficiency and to deliver more treatment that seems to be needed.
I've heard some institutions, people are leaving as early as noon because they're not having to deal with the nausea side effects. So, well, thank you so much, Dr. Thomson. Do we have some questions from our audience?
I had a question for the gallium scan. What sort of resolution do you get from, like, what size tumor might you find with from a
So
gallium scan?
with the gallium, the question was what kind of resolution, what fine detail can you see with a gallium PET scan? What's the smallest size of lesion that you could find? If the lesion is expressing a lot of somatostatin receptors on its surface, it will shine bright even if it's 3-4 millimeters in size. So the gallium PET CT will find things that might not have been seen on an indium octreotide scan just by virtue of how the 2 tracers behave and the difference between the 2 camera technologies. And so whether or not we see something with a radioactive nuclear type image is not so much a function of how finely you can slice the data, like with a CT scan. Whether or not we see something is a function of what's called target to background. So if the SUV in the lesion is 5 times the background, tiny little things will be obvious. If the SUV is similar to the background, then not so much.
Thank you for your presentation.
You're welcome.
I just recently completed, actually within the last 2 weeks, my first series of PRRT. Prior to that, I had the gallium scan and then actually a blockage in my colon, which indicated which resulted in my having surgery with a colonoscopy. So now my PRRT is over with, and I'm concerned about how it would be determined as to the success of the PRRT, and then also whether or not there's been any reduction in the size of the tumors that might allow the colon to reopen, that I could have a colostomy reversed. My oncologist indicated that actually PRRT, the effects of it could actually continue on in terms of being helpful for another 6 months or so. So any thoughts or comments in terms of how I might be able to determine my next step as far as evaluating the success of it?
So I've got thoughts that come from parenting experience in part, because my son says to me frequently, we are all special snowflakes. We are all special snowflakes. Every person is an individual, so each person's experience is going to be influenced by who they are, and their type of disease and their path is slightly different from everybody else's. There's another thing that I say back to him when he's concerned about what's around the corner, is that I really wish I had a crystal ball, and we can make projections for people based on clinical data. We can get a sixth sense of whether something's likely to go one way or the other, but none of us have the crystal ball to tell us what the natural history is going to be. And then, of course, there's the anecdotal experience that makes us all experts, and I'm not an expert because my experience is still really anecdotal, but anecdotes sometimes help a lot, and others in the audience may be able to speak to this, but there are certainly people who've had PRRT who've had not just a stabilization, but a reduction of the extent of disease that has then been sustained, not just for a few months, but potentially for years. And I have met and talked with people who have had years of feeling much better as a response to this treatment. And maybe one day Dr. Gong will be able to predict for us, just on the basis of our genes, whether or not we're going to be that person who gets the perfect response. So I don't know. But the restaging should be at least at a few weeks after the end of treatment because of the long duration of the action in the body. I would have thought at least a couple of months after the end before you would choose to look again. And the restaging generally involves the same imaging that you had at baseline so that you can compare apples to apples. And you would not want me anywhere near you as a surgeon because that's not my skill set, so I can't comment on reversing anything.
Can I ask you one other question? In terms of the colostomy, the issue of the contamination following, I was told about the urine and so on, but now you have me concerned about is that contaminating too?
Most of it's out through the urine, so it's not so much contamination any other route. It's predominantly renal and predominantly in the first few days after each treatment.
Great. I think we have time for one more question. 2 quick questions. How about you first?
Just a very quick question. Is there a wait time between the gallium and is there a wait time when you do the gallium and then when you start treatments with PRRT?
That's a really good question. Is there a wait between gallium-68 and PRRT? There is a wait time between when we give treatment and the last octreotide injection for someone who's receiving the depot injections. We can't give our treatment until it's at least 3 weeks, preferably 4 weeks after the last long-acting depot injection. So that's one of the aspects of timing. And the other is for me to be reassured that the disease is what I'm told it is. I would prefer that somebody's gallium-68 is not 3 years ago. I would prefer that it's within the last 12 months. And I tried looking to see what others do, and I'm not sure. Maybe someone in the audience can tell me what the rules are in Europe. I think maybe some centers say within the last 6 months. I think with our insurance issues for even getting that kind of scan, if I've seen it and it's definitely positive within the last 12 months, then I'm going to feel reasonably confident. The only rider to that is if somebody's got a tumor type that's not behaving like a typical neuroendocrine tumor. And we certainly did have one woman referred who had a more aggressive somatostatin receptor expressing tumor, and her indium octreotide scan went from positive to negative within the course of 4 months just with chemo. So I could not then follow up with the PRRT because the targets were not alive and expressing their receptors.
Okay, last question.
Hi. Okay, so I know with PRRT, the radionuclide originally was yttrium 90, I believe, and then it was switched over to Lutathera, lutetium. Now there's a new radionuclide, I believe, that's coming out. They're having clinical trials. It's got a shorter wavelength, but it's more targeted, and half-life is a lot, slower, or it's less of a half-life. So can you tell me about what's going on there?
I can tell you that there's going to be more than just one new option over the next decade, because this entire area of delivering radioactive materials to molecules, to targets, is just exploding. All of the novel options are only ever available in a research setting, and as you've seen with the lutetium, it takes years for our pharmaceutical industry to get something from the experiment to the clinical availability with FDA approval. A lot of the novel options also are at sites that have a cyclotron and have programs for developing novel tracers. We are a hospital with a lot of services, but we do not have a cyclotron and we do not have a team of people developing novel radioactive materials. We have 3 authorized users, and most of my job is very clinically based rather than developing novel tracers. If you want to find out what's current in the way of trials, I think the clinicaltrials.gov website is probably one of the best resources, because anything that's recruiting for a novel trial should be listed there and would give you an idea of who to contact if there was a trial with a novel agent.
Yeah, it's constantly changing, and in fact now, even for the gallium-68 scan, what's being used in Europe is something that we don't have here yet. So it's constantly evolving. You might also look at ENETS, which is the European Neuroendocrine Tumor Society, because that's a lot of the work is being done in Europe before it comes here. So
it's not completely new though, is it? 'Cause we do have 10 years, so the treatment is not brand new in concept because of the decade of experience in Europe, and that's one of the benefits of our FDA in this country being so cautious and things coming here a little bit later, is that things have hopefully been developed to a point where you know more about how well it's going to work.
The Lu-137 in combination with I think Y90 something. Yeah. Is that going to be coming here?
Y90 is used still here for our interventional radiology directed treatment targeting the liver. The Y90 and the lutetium-177, they're both delivering radioactive energy. They behave in slightly different ways in terms of how far the energy goes in the tissue and how long it stays in the tissue. And the only FDA-approved radioactive therapies that I know of that are targeting receptors commercially available are the I-131 MIBG for pheochromocytoma and this lutetium dotatate for the neuroendocrine tumors. We have a long experience with radioactive material for prostate cancer, that's a radium-223, and decades of experience with giving radioactive iodine for people with thyroid cancer.
Thank you so much. I know you're on a compressed time schedule today, so we really appreciate having you here.
Thank you.
Thank Dr. Thomson.